Haber synthesis of ammonia is the process combine hydrogens and nitrogen to form ammonia using ion catalyst. This is example of the reverse reactions and the heterogeneous catalysis. Haber process is artificial nitrogenification process and it is main industrial procedure for the productions of ammonia. And it is named after its inventor, the German chemist Fritz Haber and Karl Bosch, who developed in first decades of 20th century. In commercial productions of ammonia, the conditions is at 500 degrees Celsius and pressure between 150 and 900 atm, with the presence of the catalyst are used to give the best compromise among rates, yield and the cost of the equipment necessary to produce and contain high pressure gas at high temperature. With the absence of the catalyst, the reaction will involve via one-step mechanism and one transition state is observed. The rate of reaction is slow at room temperature. Even if we raise the temperature, it does not accelerate the above reactions, but it will decompose the ammonia molecules into nitrogen and hydrogen and lower the yield of the ammonia. However, with the presence of the catalyst, it will show the two-step mechanism or two transition state observed with the notable loss of activation energy. The dissociation of the nitrogens and hydrogen will occur and bonded to the solid catalyst surface. The hydrogen will dissociate into atom hydrogens at temperature -196 degrees Celsius, which is the boiling point of the liquid nitrogens, while nitrogens will dissociate into the nitrogens at about 500 degrees Celsius. The highly reactive nitrogen and hydrogen atoms will combine rapidly at 500 degrees Celsius to produce the desired ammonia molecules. Schematic represent the Haber process, which is first the hydrogen and nitrogen molecule will bind to the surface of the catalyst, and then the interactions weaken the covalent bonds within the molecules and eventually cause the molecule to dissociate. And then finally, the highly reactive hydrogens and nitrogen atoms combine to form a ammonia molecule, which is then leave the surface. Oswald process is the chemical process used for making nitric acid. The process is mainstream for the modern chemical industry and it provides the main raw material for the most common types of the fertilizer and pharmaceuticals. Oswald has many well-known use in both industrial and health fields. The ammonia is oxidized to form nitric acid in catalyzed chamber that contain platinum gas, which serve as a catalyst, which is oxidation is exothermic and reversible reactions. Therefore, according to Lee-Catelier principles, the degrees in the temperature will favor the reactions in forwarding directions. The first degree oxidation will produce about 95% ammonia is converts into nitric oxide. And second oxidation, which is formations of the nitrogen dioxide. Nitrogen oxide has obtained by the oxidations of ammonia is very hot. So in order to reduce its temperature, it is passed through a heat exchanger, whereas the temperature of the nitrogen oxide is reduced to 150 degrees Celsius. And then after cooling, the nitrogen oxide is transferred to another oxidizing tower at about 50 degrees Celsius and it is oxidized to nitrogen dioxide. And then the nitrogen dioxide is formed from the second oxidations is introduced into the special absorption towers. So the nitrogen dioxide will pass through the tower and water is shower the nitrogen dioxide and turn it into the form of nitric acid. Catalytic converter is an exhaust emission control devices that reduce the toxic gas like carbon monoxide, unburned hydrocarbon and nitrogen dioxide pollution in exhaust gas from an internal combustion engine into less toxic pollution by catalyzing redox reactions. Typically, there are two different catalyses in catalytic converter. One of them will tackle nitrogen oxide pollutions using a chemical process which is called as a reductions or removing of the oxygens. The other is catalyzed work by opposite chemical process which is called as oxidations or adding oxygens and turn carbon monoxide into carbon dioxide. Homogeneous catalysis. In liquid phase, the reactant and catalyst are dispersed in a single phase, which is usually liquid phase. Acid and base catalysts are the most common types of homogeneous catalysis in liquid solutions. For example, the formations of the acetic acid from the ethyl acetate, which is the presence of the catalyst like hydrochloric acid will enhance the rate of the reactions. Homogeneous catalysis in gas phase, for example, in manufacturing of sulfuric acid, which is sulfuric acid is produced from sulfur, oxygen, and water via the contact process. In the first process, the sulfur is burned to produce sulfur dioxide. Sulfur dioxide is then oxidized to produce sulfur trioxide at high temperature, which is about 550 degrees Celsius, in the presence of vanadium catalyst. Sulfur trioxide is then treated with the water, usually 97 to 98% concentrate sulfuric acid containing 2 to 3% water and forming a sulfuric acid or oleum, which is in the concentration of about 98 to 99%. This can be react safely with water to produce a concentrate of sulfuric acid. Enzyme catalysis is increase the rate of process by biological molecules and enzymes, and most enzyme generally catalysis occur at localized site called as active sites. An enzyme attracts a substrate to its active site, catalyze the chemical reactions by which the product is formed, and then allow the enzyme surface. The combination formed by an enzyme and its substrate is called as enzyme-substrate complex. The specific actions of an enzyme with a single substrate can be explained using a lock and key analogy that was first postulated in 1894 by Emil Fischer. In this analogy, the lock is the enzymes and the key is the substrate. Means that only the correct size of key or substrate can fit into the keyhole or active site of the log enzyme. The log and key models is based on the enzyme specificity, that is that the enzyme's ability to bind only the correct substrate and come from having a shape that is nearly perfect for one particular types of molecules. In that sense that the substrate fitting into the enzyme is like a key fitting into the lock. Figure shows the binding of glucose molecules in red color to the hexokinase, an enzyme in the metabolic pathway. We can see that the region at the active site closest to the ground glucose after binding. Chemists now know that the enzyme molecule, or at least its active sites, has a fair amount of structural flexibility and can modify its shape to accommodate more than one type of substrate. Enzyme kinetics shows the chemical reactions that are catalyzed by enzyme. In enzyme kinetic, the reaction rate is measured and the effect of varying the conditions of the reactions are investigated. Study is important because it helps to explain how enzyme works and it helps to predict how enzyme behave in living organisms. Enzyme activity is frequently investigated in the medicine, biochemistry and food science research field to elucidate the rates of which reaction occur and the affinity of the enzyme-substrate interactions. The rates of this reaction can be accurately measured by using UV spectrophotometer. Okay, when enzyme E bind with the substrate S, an intermediate or enzyme-substrate complex ES is produced, which is The complex which then further reacts and yield byproducts P. This byproduct can be quantified and observed by monitoring the maximum absorptions. Figure shows the rate of formations versus the substrate. From the figure, we can see that the rate rises rapidly with the increasing of the substrate concentrations. However, above a certain concentrations, all of these active sites are occupied and the reactions become zero order in the substrate. In other words, the rate remains the same even though the substrate concentration increases. At and beyond this point, the rate formations of products depends only on how fast the enzyme substrate intermediate breakdowns, not on the number of the substrate molecule present. Now we move to part 3, which is colloids. Colloids is a homogeneous non-crystalline substance consisting of large molecules or ultra-microscopic particles of one substance dispersed through a second substance. Colloids includes gel, salts and emulsions, which is the particles do not settle and cannot be separated by ordinary filtering or centrifuging like suspensions. A colloid is a mixture that has particles range from 1 to 1000 nanometers in diameters, but are still able to remain evenly distributed throughout the solutions. One way to distinguish solutions from the colloid is by Tyndall's effects, which is Tyndall's effects, also called as a Tyndall phenomenon, scatterings of beam of light by a medium containing small suspended particles like smoke or dust in the room, which is make visible a light beam entering a window. It is defined the way by which different types of mixture react to the passage of the lights focused on them. In order to understand the concept of this effect, we need to understand which of the wavelength of light are reflected and which are transmitted. Here, the light particles having a shorter wavelength are scattered, while a longer wavelength are allowed to pass through. Solutions versus colloids. Okay, we have learned about the different type of mixture, which is solutions, colloids, and suspension. They are different from each other based on the degree of the solubility of the solids in the solvent. Okay, solutions is the type of mixture in which the solid particles completely dissolve into the solvents, whilst in the case of suspensions, particles do not dissolve at all. Whilst a colloid mixture is the intermediates between the solutions and the suspensions. Its particles are too small to be seen without an ultra microscope, but are certainly bigger in the size than those of solutions. When a light passing through a colloid mixture, the light will be scattered by its particles, which is this effect is called as a Tyndall's effect. Only colloids are capable of suspending light rays that are focused on them, and no other types of mixture allow this process to occur. So this effect can be used to distinguish between types of mixture. So the solutions that allow these effects can be termed as a colloid. Colloids are on the dividing line between the solutions and heterogeneous mixture. Like the solutions, colloids can be gas, liquid or solids. Examples of each are listed in the tables. The size of the dispersed particles is the properties used to classify a mixture as a colloid. Colloid particles range in diameter from approximately 10 to 2000 angstrom, and most colloids appear cloudy or opaque unless they are very dilute. For example, homogenized milk. Okay, hydrophilic and hydrophobic colloids, which is hydrophilic colloids is a solutions containing extremely large molecules such as proteins. In the human body, the extremely large molecules that makes up such important substances as enzymes and antibodies are kept in suspensions by interaction with surrounding water's molecule. Hydrophobic colloids will not be stable in water, and the particles will clump together like droplets of oil in the water. They can be stabilized by absorption of ions on their surface, which is This absorbed ion can interact with water and stabilizing the colloid. The electrostatic repulsions between the particles prevent them from clumping together. So this can be also stabilized by the presence of other hydrophobic groups on their surface. For example, sodium stearate or STOP molecule has a polar head and a long hydrocarbon tails that is non-polar. So the actions of the STOP is act as a dual nature of the hydrophobic tails and the hydrophilic ends group. The hydrocarbon tails is soluble in oily substance which is non-polar, whilst the ionics or carbonyl groups remain outside the oily surface. Figure 1 shows the hydrophilic groups on the surface of a giant molecules that help to keep the molecule suspended in water. Figure 2 shows the schematic illustrations of the stabilizations of the hydrophobic colloids in water by adsorbed ion, which is hydrophobic colloids can be prepared in water only if they are stabilized in some way. Otherwise, their natural lack of affinity for water causes them to separate from the water. Hydrophobic colloid can be stabilized by absorption of ions on their surface, which is this absorbed ion can interact with water and stabilizing the colloids. At the same time, the mutual repulsions between the colloid particle with absorbed ion of the same charge keep the particle from colliding and so getting larger. Figure 3 shows an emulsion of oils in water by stearate ion. The additions of the sodium stearates or any similar substance having one end that is hydrophilic, which is polar, and hydrophobic, which is non-polar, will stabilize the suspensions of oil in water, which is the hydrophobic ends of stearate will react with the oils, and the hydrophilic head end point will react or interact with water.